what are we doing? We're doing biology. All right. It's your boy. It's your boy. It's your boy. Put it into pure math, right? Should be. Huh? Why were you looking at yesterday? Oh, okay. What is this, bro? What is this? It's cool, I guess, but it's kind of confusing. Okay, yeah, I did put into pure math. Okay. Okay. So, what are we doing? We're trying to complete 5A. What did we do yesterday? What did we leave off? We left off talking about autotrophs and heterotrophs and chemotrophs, and we didn't talk about the different types of chlorophyll. We did talk about a little bit about how the electron transfer chain worked, but that was kind of getting besides the point. We went pretty deep into that actually. Let's just erase this. Okay, I shouldn't be doing maths in a long time, which kind of sucks, but I guess we could do it on the bus or something, because it is pretty low effort. You know what I really want to get? I want to get, like a tablet that I could, like, write on. I really want to get something like that. That would, like, really, really do me a lot of good. Okay, we can save this as well. What am I looking for? I'm looking for my notes. Okay. We should probably print those notes down. Should we? I feel like it could help. Maybe. Let's see. I mean, yeah, it's not even that many pages. We should really print these. Okay. Yep, so this is where we left off, right here. This talks about everything, right? Light independent, where's light dependent? Okay. Okay, here you go. Yeah, the two things. They don't have the picture. Okay, they just skipped the electron transfer chain. Interesting. Then they talk about limiting factors, which, like, fine, I guess. Okay, that's fine. So we'll just go.
You don't have mechanics anymore, right?
I have no idea what the school is planning to do. Literally, I'm জানি না.
তুমি জানো না, জানার চেষ্টা করছো? তুমি জানো না বলে
I
থাকছো, তাই না?
who am I supposed to ask? No one I know knows either. The school decides whether this is weird. Even I'm confused. Apparently, the school decides whether I can take mechanics next year or not, and which doesn't seem right.
মেকানিক্সের জন্য অপশন কি তোমার এসে ছিল? তুমি নিজে চুজ করছিলা মেকানিক্স?
That was for AS, not A2, not this year.
Why did you take this subject? তুমি জানতা নিশ্চয়ই যে এই সাবজেক্টে গ্রেড ভালো আসে না? ডিড ইউ মানে কি বলবো অ্যানালাইসিস করেছিলা কোনো? খোঁজ নিয়েছিলা যে সাবজেক্টে গ্রেড কেমন আসে মেকানিক্স?
I mean, I did it throughout the year. I never had that many problems with it. So, anyways,
যেটা হয়ে গেছে বাদ দাও। হ্যাঁ, বলো কি? আমি জাস্ট জিজ্ঞেস করছি, তুমি জানো না, ওকে, স্কুল খুললে খোঁজ নিয়ে জানবা। আমি পড়ো।
there's a chance যে আমি statistics করবো, and ওটাতে maybe আরো খারাপ হয়ে যাবে. I don't know. Hopefully not. Then there's also a chance we can take mechanics too, which is just the next part of mechanics.
মেকানিক্স তুমি অল অফ আ সাডেন কিভাবে লেভেলে নিবা? নাকি স্ট্যাটিস্টিক্স? ডেফিনেটলি না।
That happens.
So they will give me, huh?
That happens.
মানে অপশন আছে দেওয়ার?
No, it's like last year কি হয়েছিল? The first batch of students took mechanics. See, the students who took mechanics and statistics.
Mm.
The students who took mechanics, they did pretty bad. So the next year they took statistics one, which is the one that the other.
They took তার মানে অপশন আছে
No, the school said we're not teaching mechanics two. We're teaching. We're going to be. We will either teach you statistics one, or you study mechanics two at home. I'm guessing, and then maybe we can just give you the paper. But we're not going to be offering classes for mechanics two. এবার বলছিল যে if at least 10 students get an A for mechanics, then we will teach the mechanics two in the second year. But I don't think.
তার মানে যে এই সাবজেক্টটা আসলেই কঠিন একটা সাবজেক্ট তুমি তাহলে জানতা এটা you couldn't drop it and take the other subject
Well, I mean, অনেকবার শুনছি that the other subject was even harder, but then I don't know. And anyway, I mean, I never specifically thought that.
তার মানে
Okay, I'm more like every other
তাদেরকে স্ট্যাটিস্টিক্স দিয়ে দিবে এই তো
yeah, yes, yeah.
তার মানে মেকানিক্স থেকে স্ট্যাটিস্টিক্স দিয়ে দিয়ে doesn't that make sense আমরা যারা ভালো করছিল ম্যাথে তাদেরকে ওরা মেকানিক্স অফার করছিল or maybe you chose mechanics in the AS
I literally could not drop mechanics. I literally asked my teacher. They said because I took physics, I had to take mechanics.
oh my god now you're dropping physics anyway যেটা হয়ে গেছে বাদ দাও যা দিবে দিবে তোমার তো করার কিছু নাই না you have no choice in A-level
Unless I really want to take mechanics too, which I don't really know.
D পাচ্ছ তো তুমি কেন এটাতে যাবা তুমি আস্ক করবা টিচারদেরকে যে what should I do is statistics even harder for me will be even harder for me then math টিচার আছে এতদিন থেকে তুমি বলতেছো হ্যাঁ I will talk to this teacher 3 মাসে তুমি এই কোশ্চেনটার অ্যানসার নিতে পারো না টিচারটার কাছ থেকে
Why would who ask what?
টিচারকে জিজ্ঞেস করো তুমি যে ম্যাথ করো যে টিচারটার কাছে যে আমি এটাতে ডি পাইছি শুড আই ড্রপ দিস মেকানিক্স দুইবারই আমি ডি পাইছি আমাকে এইটাকে আমি ড্রপ করব করে আমি কি স্ট্যাটিস্টিক্স নিব বিকজ देयर इज আ পসিবিলিটি যে ওরা আমাকে স্ট্যাটিস্টিক্স দিবে শুড আই টেক ইট অর আমি বলবো যে না আমি মেকানিক্সে থাকবো হোয়াট শুড আই ডু ডিসকাস করা যায় না শি টিচেস অল দ্য সাবজেক্ট রাইট
I don't think so. I don't think she teaches statistics.
আচ্ছা ডিসকাস করা তো যেতে পারে না এনিওয়ে দেখো
I'll probably take statistics anyway, so about that. Did we have to talk about that?
wow no more Saturday class with Ustad wow বলে না বলে এগুলো আল্লাহ
Can you turn on the AC, please?
তুমি উঠে যেতে দাও
কারণ আমি পড়াটা শুরু করি এভাবে এসব কথা
ওকে সরি সরি বন্ধু
Okay, so when we talk about making energy from organic compounds, we use carbon dioxide and water. Where does carbon dioxide? Okay. Did I say they make carbon dioxide because of photosynthesis? No, they turn carbon dioxide into organics. Carbon dioxide is not the organic. Oh my god, that's embarrassing. It's more like energy from organics, from CO2 through photosynthesis, and the energy for this and the energy which actually comes, the energy inside this comes from the energy that's inputted via photosynthesis, which is coming from the sun. Okay. Yeah, that's pretty much it. I don't know what else to really bring up. I think we can make this a bit thinner. Yeah, okay. So yeah. This drawing pad is actually really big, honestly. Could be bigger, but it is still pretty big. This is just the chemical equation. Photosynthesis is a process used to convert carbon dioxide and water into organic glucose using energy trapped from chlorophyll from sunlight. So we talk about the energy coming from the sun, and it's used in photosynthesis. Well, how is that energy trapped? It's by the chlorophyll pigments. Yeah. So we have chloroplast structure and function. Chlorophyll is the pigment which, or is chloroplast that pigment? Yeah, there you go. So chlorophyll is the primary photosynthetic pigment within the chloroplast. Chlorophyll absorbs red and blue light from the sun, converting it into chemical energy, but chloroplast is a part where the photosynthesis takes place. So you have the chlorophyll pigment, which absorbs the sunlight, but where the photosynthesis takes place is going to be the chloroplast. Okay. Okay. Is there anything I want to do before we continue? I don't think so. So here we go. So when we talk about the chloroplast, okay. First thing is, where is like the nucleus substitute, right? So there you have the DNA loop, kind of similar to a prokaryotic thingy, a prokaryotic organism, right? You have your DNA loops, kind of like plasmids and stuff. Starch grains for products of photosynthesis. Are starch grains and ameloblast the same thing? I don't quite remember. So it's a double membrane organelle. So you have the inner and outer membrane. The outer membrane is going to be freely permeable to molecules such as CO2 and H2O, which is, conveniently the two reactants for photosynthesis. Inner membrane contains many different transporter molecules. These are membrane proteins which regulate the passage of substances in and out of the chloroplast, and these substances include sugars and proteins synthesized in the cytoplasm of the cell but used within the chloroplast. The inner and outer chloroplast membrane form an envelope. Sugars and proteins synthesized in the cytoplasm of the cell but used within the chloroplast. Right? Now, I'm not aware of any proteins that are used. I don't know what NADH is. Is NADH a sugar or something? I'm not really sure. Okay. Then you have thylakoid membrane, thylakoid space, and granum, as well as stroma. Stroma is going to be kind of the cytoplasm of the thing. It's the fluid that surrounds the thylakoid membrane. Thylakoid membrane being the membrane of these structures inside. So it contains all the enzymes needed to carry out the light-independent reactions of photosynthesis. Now, since these are covered with the chlorophyll pigment, you can kind of tell that over here is where the light-dependent reactions are going to be taking place, while the light-independent reactions are going to be taking place in spots without any chlorophyll, which is going to be the stroma. Okay, so yeah, now the thylakoid membrane is going to be a system of interconnected flattened fluid-filled sacs. Okay, so similar to the mitochondria where you had sacs which were doing most of your work, over here once again we have sacs of fluid-filled system of flattened fluid-filled sacs. Okay. So what's special? Proteins including the photosynthetic pigment of chlorophyll and electron carriers are embedded in the membranes and are involved in the light-dependent reactions. So, as you're aware, for the light-dependent reactions, we need the electron transfer chain, which means that we need electron carriers, and we need those transport proteins on the thylakoid membrane itself to actually pump in and pump out the NADH and the H plus ions. Sorry, not the NADH. The H plus ions from the stroma into the thylakoid membrane. So you can kind of imagine that the thylakoid membrane is going to be having most of these. Similar to how the stroma, it's going to be containing most of the enzymes that are needed for their own reactions. Now protein, know that if they are used, they're going to be embedded in your membranes, and know that you already know that they're going to be involved in your LID reactions. Okay. Now you have spaces between your thylakoid membranes, right? And that's going to be another fluid within your membranes, sacs. And those are going to be containing enzymes specifically for photolysis, okay? So, photolysis. I need to learn more about photolysis. But okay. Yeah. It's gonna be, that's kind of where photolysis takes place in the thylakoid space. Now in the granum, is a stack of thylakoids joined to one another. So you can call that thylakoid a singular sac, whereas a granum is going to be a stack of those sacs joined one to another. And the tube is called, what is it actually? There are names for the tube for the separate stuff. A thylakoid is a single sac. A granum is a stack of thylakoids. Wow, that's a tongue twister. Is that really it? That's all we really need to know. Okay. Cool. So, okay. Which contains many transport molecules. These are membrane processes which regulate the passage of substances. Yeah. Stack of thylakoids joined to one another. Grana resemble stacks of coins. So grana is the plural of granum. Okay, cool. So, just some trivia real quick. The stroma is where you have LIR. The thylakoid space is where you have photolysis. The thylakoid membrane is where you're going to have LDR. So therefore, photolysis isn't exactly... Is photolysis... do you need energy from sunlight for photolysis? Or is that something that happens separately? I don't have memory of that. No, right? Does photolysis require energy? Does photolysis and the conversion of NADH to NAD to NADP to NADPH require energy the same way require energy from sunlight? Photolysis split the water molecules using light energy. The energy from the sunlight is absorbed by photosystem 2 which excites electrons to high level. These high energy electrons are passed on, but the photosystem needs to replace them. It obtains replacement electrons by splitting. This reaction is this. The energy absorbed by PS2 from sunlight is what drives this process. But no. For photolysis, you say that the hole left by the electron that leaves is caused by the is caused by energy from the sun. However, is the process that can drive the photolysis to occur, but does the photolysis reaction itself require energy? And I'm just gonna disable this. Is it on or off? It's off. Okay. To fill this electron hole and stabilize the photosystem, an enzyme complex associated with PS2 splits a water molecule. The energy from the absorbed photon is not used for just one or the other, but rather a chain of events: light absorption and electron excitation. A chlorophyll molecule with photosystem 2 absorbs a photon of light. This light energy is immediately transferred to electron, boosting it to a very high energy state. The high energy electron is so unstable that it is ejected from the PS2 reaction center, is passed to the first electron acceptor in the electron transport chain. The ejection of the leaves a positively charged unstable hole. To fill this hole and restabilize the PS2, an enzyme complex associated with PS2. This is the energy from the sun that created the hole in the first place. What an oh. Creation of a powerful oxidant. When a photon of light strikes the special chlorophyll molecule, an electron is excited and ejected. This leaves the chlorophyll molecule with a powerful positive charge, often written as P680 plus. This P680 state is an incredibly strong oxidizing agent, the strongest known in biology. What? Its redox potential is high enough to strip an electron from even a very stable molecule like water. The energy from the sun has now been converted into a high energy unstable charge separation P680 in the ejected electron. The P680 plus acts as the pull that extracts electrons from water. The actual splitting of water is catalyzed by a protein complex called the OEC, which is a part of PS2. This complex contains a cluster of manganese and calcium ions that facilitate the breaking of water bonds and the transfer of P680 plus. So light energy doesn't directly split the water, it creates an extreme chemical potential electron hole that makes the splitting of water a thermodynamically favorable reaction, allowing the enzyme complex to perform this difficult task. Apparently, okay, wait 1 second. Okay. So that's not a very good link. Do Br- 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 Okay, I think we're good. That explains it. Okay, and for okay, that was for photolysis, and for conversion of NADP plus to NADPH is the final step in the linear electron flow of the light-dependent reactions. So obviously it requires energy, right? You need the photon of light to hit the PS1 for the electron to be moving towards this. Um, mm, 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 mm. Now, in this, the electron just goes back into the chlorophyll. In this, the electron goes into NADP. Now, in the example that we were looking at earlier, we actually kind of skipped photosystem one, because it said electron transfer chain, make your ATP, and then the electron immediately goes into NADP to make NADPH. That was in that secondary photosystem, and I'm not really sure what this photosystem really does. I guess since it's out of energy. No. No, that electron is what fills the hole for the second one to go in, right? But then there is no electron transport chain here. So this is a different story. Why is there a second electron needing to be emitted? Why can't this electron be the one that goes into NADP? Why is the electron going from photosystem 1, going from photosystem 2, go into photosystem 1? Why can't that electron go straight into NADP in one swoop? Why is there a need to use an entirely separate photosystem in order to send an electron to NADP to make it to reduce it? Also, I've seen diagrams of electrons being emitted from a photosystem, undergo the ETC, then immediately go into reducing an NADP. However, that is not happening here. The short answer is one photosystem alone does not provide enough energy to reduce NADP+. You need the combined energy boosts from both photosystem 1 and photosystem 2. Energy requirements for reducing NADP+. NADP+ is a relatively high energy electron acceptor. You need electrons that are really energized to reduce it. The photons exciting PS2 raise electrons' energy level partway enough to drive them down the ETC and pump protons for ATP synthesis. But after moving to the ATC, the electrons have lost a chunk of that energy, and by the time they reach the end of PS2, electron transfer chain, they're not energetic enough to reduce NADP plus directly. Why not go from PS2 to NADP directly? If PS2 went directly to NADP plus, it's NADP plus to NADPH. Sorry. Okay. You'd get little or no ATP from phosphorylation, from photophosphorylation. What is photophosphorylation? Because the electron transfer. What is photophosphorylation? Oh, okay. It's this entire process. You'd get no energy from photophosphorylation because the ETC proton pumping step would be bypassed. The electrons wouldn't have enough energy left to actually perform NADP plus NADPH reduction efficiently. Role of photosystem 1. Photosystem 1 re-energizes. Wait. But again, there's no NADP mentioned here, right? Yeah, so it can't be cyclic either. Unless these electrons can go to NADP, however, what's going to be refilling them, right? Because there's nothing going on here, so. And also, they can't go to NADP because then they would lose out on the energy. Like they don't have enough energy to go back to NADP to go to NADP. So role of PS1. PS1 re-energizes those electrons with a second photon hit. Now the electrons are boosted enough to have a much higher energy level again. Well, that's not true, is it? Photons hit the PS1 and new energetic electrons are sent, and the old electrons from the ETC refill the holes left by them. Oh, well, okay, I suppose that, see, they're coming to fill in the holes left by that. But by the time the next ones come, those are the electrons which are sent out again, so I guess it's fine. Yeah, okay, I guess it's fine, yeah, okay. Why do some diagrams show one photosystem directly reducing NADP? That's often cyclic phosphorylation diagram simplified, or bacterial photosynthesis systems with only one photosystem. What is that sentence? In plants, the full non-cyclic phosphorylation path uses PS2, ETC, PS1, NAB+. The single photosystem to NADP versions or simplifications are referred to systems where the photosystem's excitation energy is sufficient for the electron to jump straight to NADP+ without a second boost, but that's not the case in plant chloroplast Z scheme. So true, so true, so true, so true, so true. I can explain where the brand new electrons idea come from. It's from cyclic phosphorylation, which look different from this non-cyclic setup. No, I guess it's fine. No constant brand new electrons. PS1 doesn't get a fresh batch from water like PS2 does. It depends on PS2's Electron to sort of replace the ones it loses 1080 plus. Yeah, so true, so true. Yep, that's true. Yeah. Damn. Damn, boy. Sorry. Damn, boy. Boy. Meow, 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 meow. Do, 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 do. Ba, ba, wah, 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 Yes, let's see. Is there a way I can start using GPT? What's that? 3.5, whatever. Do I really?
নামাজে যখন তোমার মধ্যে থামাথামি কি আছে বাবা?
Um. All right. I will be back. Okay.
তাহলে ফ্রাইডে স্যাটারডে সানডে 3 দিন টানা নিয়ে নাও।
Friday, Saturday, Sunday? No, I want gaps between. Wouldn't that be better?
ওকে, তাহলে ফ্রাইডে সানডে and
Tuesday.
one day in between.
Friday, Sunday, Sunday, Monday, Tuesday. Friday, Sunday, Tuesday.
Friday, Sunday, Tuesday.
Welcome back, everybody. Welcome back. Welcome back. Little bra, insane. Yep. Alright, how are we doing? Let me just set up something. 13.k research. My farming is fine, but maybe as I grow, I'll need more. Can we get more Wardenclyffes? Okay, we can. Should we get this? I mean, why not? This and this. What are we slow on? Aluminium. We are slow on. We are pretty slow on cement and iron. We're doing okay. Okay, so cement worker will increase. What's up? I'm just setting up one thing before I stop.
Faizan,
Yes. Did I do Wardenclyffe? Then I'll put one to this.
বিসমিল্লাহ কোন সময় তোমার পড়া হয় না? বিসমিল্লাহতে এগুলাই হইতে হয় সব সময়?
6.
যে জিনিসে তুমি বের হইতেই পারো না, এরপরে এটাই বিসমিল্লাহতে থাকতে হবে না? ও কথা হচ্ছে শোনো, সত্যি কথা না মিথ্যা কথা বলছি, বলো।
It's not true. Knowledge gain, each side is 0.95. I think we can actually convert most of these into this and just leave it at this. Okay, is there anything else? Save it for the next one. I don't really need this. Are we full on that? Oh, we are. Okay, can we convert? Okay, it's already on this, so maybe another smelter would it be good? What are we missing from that? Iron. Our iron cap is blocked. Okay, so maybe we can get some more crates, construct a few, put them into iron. So now we can get a new smelter. Honestly, a few more smelters would be nice, because then we can improve our iron thing. But then coal is gonna be another block, because our coal isn't great either. Okay, essentially we're just waiting for. Okay, we'll just make one more farm and we'll put that into coal mine. Okay, we'll put that into coal mine because coal mine is good because coal mine or cement plants. Coal mine is good because if you put them into coal mine, that kind of gives us some room to work on our smelter, which gives us iron and aluminium, which both we're full on right now, but yeah, our food is literally perfect, so I'm not gonna worry about that. We're gonna spend a little bit more money on this, and we're going to kind of cap on some crates. How much do we have? We can make 9. Just 1, 2, 3, 4, 5. And we'll spend this on no, we're gonna spend this on steel a few times. Actually, just once. Coal, definitely. Aluminium. And food. Do we need to put on food? I don't think we need to put on food. We'll put it on. We'll put on copper. Okay, and for research, what are we missing? We're missing knowledge. Knowledge. No, we're not missing knowledge. We're fine on knowledge. We're missing steel. Steel 2.5, that's fine. Coal 2k, that's fine. Increases iron output of smelters by 20%. That's gonna be crazy good. Discover electricity, no quiets required. That's also gonna be really good. Let's do this. So our iron should be really good now. 1.87. What's going on? Smelter 12%. Okay, whatever. So next it's going to be electricity. So we'll just put this on. Steel beams, reduce cost of cabins. Saving up on steels. This in costs. Okay. Yep. That's what we're gonna leave it on. Okay, cool. Ba-ba-da-ba-da. Nine. let's start once again. Lettuce. Do, 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 do. Amma, could you give me my glove from there? When will I like do something else? Actually, I don't know. Okay. What the? It's okay. Do. Okay, fine. So I need to speed up. So since we already know that's double membrane, space between the inner and outer is known as the chloroplast envelope, like similar to the nuclear envelope. System of membrane disk is known as thylakoid. We already know this. Stack of thylakoid is grana. Existing stack layer increases surface area for more light absorption, so that's why they're in stacks. Because remember, in a grana, okay, the surface, the thylakoid membrane, is where the LIR reactions happen, which means you have your photosystems over there, which means that you have your chlorophyll over there, your chlorophyll pigment over there. Therefore, you want the max surface area over there. And the inside is where you have the photolysis going on, which also requires light. But that light is again really just that energy. Well, that energy is really coming from the photosystems. So yeah. Thylakoid membrane are a site of light-dependent reactions. Yep. They contain a photosystem, electron carrier proteins. Thylakoid space provides space for H+ ions that's required for the electron transfer chain. Grana are joined together by lamellae. Okay, there you go. That's the tube, right? So the extension of thylakoid membrane is called, or the tubes are called lamellae, which maintain proper distance between grana to maximize light absorption. So that's the function of these lamellae. So to spread these grana apart, these grana towers apart, enough so that maybe one is not shading the other and it has maximum light absorption. This is pretty cool. Stroma is the site of light-independent reactions, as it contains enzymes like Rubisco involved in Calvin cycle. This is the only enzyme that I'm aware. This is the only enzyme that I'm aware that they actually mention that takes part in the Calvin cycle. I actually didn't even know it was a Rubisco. It was an enzyme. Sorry. Anyways, all I know is that it contains carbon, and it comes from GALP as well. Chlorophyll is, here we have the pigment, chlorophyll, synthetic photosynthetic pigment. It's not a single pigment, but it is a mixture of closely related pigments. So you have A, B, carotenoids, and phaeophytin. So you have blue, green, yellow, green, orange carotene, yellow xanthophyll, gray pigment. Okay, so it is the, one second. Okay. So yeah, AB carotenoids, which contains carotene and xanthophyll and phaeophytin. So A, B carotenoids and phaeophytin. This contains your carotene and xanthophyll, and this is again just phaeophytin. Remember the colors. This is blue green. This is yellow green. This is going to be orange, and this is going to be yellow, and this is going to be gray. Okay. Carotene and xanthophyll carotenoids are known as accessory pigments because they absorb and pass on energy from light wavelengths that are not efficiently absorbed by primary pigment chlorophyll A. So the main wavelengths. So okay, so okay, why only specifically A, not B? Okay, so what we can say for sure is that the carotenoids are known as accessory pigments because they absorb the light wavelengths that primary pigments like chlorophyll A do not absorb. Okay. Photosystems are a collection. Okay, so this is a definition of photosystem. They're a collection of chlorophyll, which is a primary pigment. Accessory pigments, it's a collection of chlorophyll, which is your primary pigment. So therefore, these are primary pigments. Primary pigments, chlorophyll. Okay. So your chlorophyll means it's a primary pigment. So you have your chlorophyll, you have your accessory pigments, you have your proteins. Of course, your proteins are needed for like your transport proteins and your carrier, your transport proteins inside your membrane, your carrier proteins. Okay, well, the photosystems are collection all within a membrane protein. That means the carrier proteins are not involved. It's just the sorry. The transport proteins are not involved. It's just the carrier proteins. The carrier proteins which carry your electrons, but not the transfer protein. It's called carrier protein or transfer. Transfer proteins are the ones inside here which the electron does redox reactions and one after another, and then leaves, and that actually pumps in hydrogen into the thylakoid membrane. So into the not thylakoid membrane, thylakoid space. Yeah. So yeah, that's not what we're talking about. We're talking about the other thing. But we need to remember there's two types of photosystems, and they differ only between really the how much light or the light absorption peak that they have. So over here 700, over here 680. This is known as P70 because they have this, and this is known as P680 because they have this. Okay. Absorption and action spectra. Okay, so again, photosystems, collection of your chlorophyll, your like secondary chlorophyll almost, and your proteins that are involved in carrying your ejected electron around, right? Absorption action spectra, which is how these two differ in this. Do these differ because they have different chlorophyll ratios? Let's see. Does PS1 and PS2 differ in absorption peaks because they have different ratios of chlorophyll and chlorophyll pigments? It's not just the ratio, but it's the specific types of chlorophyll and accessory pigments bound in each. PS2 has a special pair of chlorophyll A called P680. PS1 has P700. The difference comes in slight structural differences in the protein environment surrounding each chlorophyll pair. The proteins subtly alter the energy levels Both they contain chlorophyll A, chlorophyll B carotenoids. The pigment ratio and an arrangement in the light harvesting, which affects the thingies, which affects the range of wavelengths absorbed and how efficiently energy is transferred. Why the difference matters? Because these are at different energy levels. PS2 absorbs electrons at a higher redox potential than PS1 does after its excitation. PS2 needs higher energy photons than PS1 so that they can split the water. PS1 can use slightly longer because it's still working with electrons that came from PS2 with some energy already. Cool. Okay. So I'm gonna write this down. Photolysis and NADP plus to NADPH both require light energy. However, the amount required differs mainly since the electrons emitted from PS1 came from PS2 with some energy already. So PS1 uses longer wavelength, less energy. PS2 needs more energy for photolysis plus etc. Okay. Okay, so again, how do they differ? They differ because, again, remember that they in its core, the reaction center has different absorption peaks. Okay, they have different proteins. Well, see, since it's a collection of primary pigments and accessory pigments, um, let's see, slight structural changes in the protein environment surrounding each chlorophyll pair. The proteins subtly alter the energy levels of the chlorophyll molecules. It's not just the ratio of pigments, but the specific types of chlorophyll and accessory pigments abound. So PS2 has a special pair of chlorophyll A molecules called P680, and PS1 has a special pair of P700. So that by itself is a reason for why they're different. Okay. Anyways, I'm not gonna go too much into it. So, absorption spectrum shows the absorption of different wavelengths by different photosynthetic pigments. Action spectrum shows the rate of photosynthesis at each different wavelength. So they're shooting specific wavelengths of light at it and seeing at which wavelength of light is the most oxygen produced, is the most C6H12O6 produced. Because at the end of the day, you know that the light dependent reactions are the main thing that hold LIR. So if there's LDR at a smooth rate, you're gonna have LIR at smooth rates. So over here we can see that the relative absorption of light, you can see that it's best over here, kind of right before this 500 and if this is 550, this is more like 560, 570 almost. So before 570 is where you have your peaks, but you have rises and falls everywhere. You can see in between where the chlorophyll A and B fall, your carotenoids also have that peak. So over here where chlorophyll A falls, no, this is chlorophyll B, but wait, where's carotenoids? This is carotenoids, right? This is carotenoids, this is chlorophyll B, this is chlorophyll A. So you can see where chlorophyll A falls, carotenoid goes up. You see where chlorophyll B falls, carotenoid goes up as well. Over here, chlorophyll A falls, so carotenoids kind of picks up here as well. I think that's chlorophyll. I think that's carotenoid. Is that carotenoid? Okay, never mind. That's chlorophyll B. So it's mostly here, I suppose. But over here, relative absorption of light, full absorption, rate of photosynthesis on the right. So you can see, yeah, as light increases, photosynthesis increases. So it's okay if they're on the same y-axis. So you can see that in this area on the left side you have pretty good photosynthesis. On the right side you have some, but there is a plateau in between and on the right side. Full action spectrum of chlorophyll. Full absorption spectrum of chlorophyll. What is the difference? Is action as in photosynthesis? Action spectrum shows rate of photosynthesis. Okay, so the red is the rate of photosynthesis and the black is the rate of absorption. Cool. And you can see how closely they're linked. So at these wavelengths, we put these wavelengths in and we measured how much light was absorbed, and we also measured how much rate of photosynthesis was there, and we did that for all these, and you can see how closely they're linked. And this is proof that the pigment is used in photosynthesis, as the cumulative absorption spectra produces a similar graph to the action spectra. So they're obviously linked together or we could say the absorption spectra, or we could say what is the action spectra? It shows the rate of photosynthesis. What is the absorption spectra? The amount of light absorbed. So therefore, we can say that the rate of photosynthesis is joined together to is linked with the light absorbed. So there's a correlation between the absorption and action spectra, as they both have peaks and troughs at similar wavelengths. Advantages of having different pigments. So since you have each pigment absorbing a different wavelength of light and absorbing different amounts of light at each wavelength, a greater range of wavelengths is going to be able to be absorbed, and a faster rate of photosynthesis can be maintained. I don't know if this is necessarily true. Some pigments have protective role in light harvesting, such as accessory pigments. So so they're really describing why do we have pigments at different which do best in different pigments, so that when we have ratios of different pigments, we can or when we have collections in photosynthesis of different pigments and accessory pigments, we can have a greater range of wavelengths. And if if each of them absorbed their own peak at different amounts of their own peak of light at different wavelengths, then we can actually have a greater amount of light absorbed at each wavelength, allowing a maximum or very fast rate of photosynthesis at multiple wavelengths. Because light is not a controlled thing. Light can come in many different wavelengths. So it's the plant's job to be able to say, hey, we can absorb light at this wavelength, at this wavelength, at the best absorptions as well, so that we can have max rate of photosynthesis at all wavelengths. Okay, chromatography for separation and identification of chemicals such as photosynthetic pigments. So okay, what are we doing here? So we're grinding leaves into propanone and collecting filtrate loaded onto chromatography paper and placed into solvent. Solvent is below baseline. Obtain solvent front and determine R value. R values are specific to pigments. Okay, here we go. So R value is specific to pigments at a specific solvent. So it's important to use the same solvent so that you can compare. Different solvents permeate some membrane of chloroplasts differently due to different permeability. Chlorophylls have different solubility in different solvents due to their different structures. So we can identify what kind of chlorophyll pigment is used in a leaf by doing chromatography and comparing it to like already pre-recorded, like a data book or something. Yeah, okay, cool. And the reason, okay, and these are the two reasons why we can use a data book, because each chlorophyll has a specific value for each solvent, because different solvents permeate, different solvents permeate some membranes of chloroplasts differently due to different permeability. This doesn't... See, remember when we said the cell membrane of chloroplast? What is the cell membrane of chloroplast? That's obviously the... you have the inner and outer membrane, right? And we said that the inner membrane was permeable to H2O and CO2, remember? So obviously when you're talking about different solvents like ethanol and propanone, right? They're going to have different structures, and so therefore they're going to have different permeabilities going through the two membranes, right? The double membrane envelope, right? And so they're going to have different permeability. So therefore, these chlorophylls are going to have different solubility, right? Does that make sense? In different solvents, they're going to have different solubility. And not only that, but it's going to differ from chlorophyll to chlorophyll because each chlorophyll has different structures. Now we haven't really mentioned how each chlorophyll can have different structures. But are we talking about chloroplast here or chlorophyll? Because when we're talking about chloroplast, then I can understand the membrane, but I don't understand how chloroplast can have different structures. However, chlorophyll can have different structures, but chlorophyll has its own membrane? Ha- I suppose. No. Wait, no. See, permeate some. Oh no, okay. Whoops. Never mind. Chloroplast. So they penetrate the chloroplast. So how they penetrate the chloroplast can differ depending on the solvent, but how the chloroplast differs in how it's getting dissolved depends on the chlorophyll. So over here, it's different structure, struct of chlorophyll, and this is struct almost of solvent. Biochemistry of photosynthesis it takes place in two stages. You have your light independent and light dependent. This is not dark stage because light independent stage. Light independent stage can take place in both presence of light and absence. Obviously, this occurs light dependent in the thylakoid membrane. Part of it happens in the photolysis. Photolysis, which happens in the thylakoid space. Break down water molecules in photolysis to provide H plus ions for reduction processes. Reduction processes to turn NADP plus to NADPH. Produce ATP via electron transport chain needed for light independent reaction, aka ATP to ADP, especially the coupling reaction to turn was it 3GP into GALP, I believe. I don't remember it quite well. But over there you also need you also have this following this reverse reaction in the same process, because it needs something. I think it needs the hydrogen in the GALP for it to occur, and you can figure that out by looking at the structure of GALP. So photon of light accepts electrons, pushes the electrons to a higher energy level. If it gets excited enough, it'll bounce around to different carrier proteins until it goes into a specific one, I believe, and that's going to be your electron carrier protein. And so via the electron carrier protein, they leave the chlorophyll, and so they're moved into that electron transfer chain, which brings hydrogen ions from the stroma into the thylakoid space, and then they finally go into and finally the electron gets. Oops, hold on. Okay, yeah. So the electron then moves on to the PS2. However, PS1, sorry. However, after the hydrogen ions are moved into the thylakoid space, they move out into the stroma once again through a pump, which then turns ADP plus PI into ATP. And so this whole process differs by one thing and one thing only. First of all, if this is going to be PS, this is always going to be PS2, right? Or, okay, this is only PS1. Okay, so this, okay, so what we talked about right here, how this electron transport chain worked to make ATP, can be similar in both cyclic and non-cyclic phosphorylation. What's the difference in both? So both phosphorylations happen at the same time. Remember that why does I remember cyclic phosphorylation only occurs because it doesn't get bottlenecked by ADP and NADPH. So so that's exactly the reason, because because it doesn't get bottlenecked by NADPH and can still produce ATP. Both produces ATP by chemiosmosis, but only non-cyclic produces reduced NADP. So phosphorylation, the only difference is this chlorophyll is in the PS1. This chlorophyll starts from the PS2, and so it goes, light hits it, it ejects an electron, it gets accepted by electron carrier protein, goes down the electron transport chain, makes ATP, comes back to PS1 and gets excited the same exact way. Both, so the photons hit PS1, excite electrons, they get accepted by electron carrier protein, move to ETC, electron transport chain, and then the PS1. So, okay, now listen. After, one thing you need to know, since it's called non-cyclic, you have to know that the chlorophyll, the electron that leaves the PS, in that case it's PS2, but when they leave the chlorophyll, they never come back to the chlorophyll. For both electrons in PS1 and PS2, what happens is then there's a hole left that needs to be filled by another electron. So the photolysis of water releases electrons that replace the electrons lost in PS2. This photolysis doesn't happen automatically. The hole, I can't just say apparently. I need to know if this is true or not. So the hole has some. It's positively charged, and so it kind of attracts the it kind of attracts the photolysis to occur, and moves and attracts the electron to be fill that hole left. And so now that it fills that hole, electrons from PS1 are also accepted by electron carrier and ATP, which also accepts H plus from. Okay, so NADP is also another electron carrier, but NADP accepts the H plus as well from the photolysis of water to become reduced NADP. So NADP you can think of as another electron carrier, but it accepts that hydrogen ion as well to form this reduced NADP, which again, that H is a primary thing to form that gallop from 3GPa or 3GP or whatever it's called. So this is your equation for photolysis. I'm pretty sure you're supposed to remember this. So this is hydrogen ions is going to be used for the NADP. This O2 is lost from lead. This is a waste for the plant, but it's obviously what keeps us alive. And this obviously requires energy in the form of, I have no idea. Let's see. Let's just ask ChatGPT. Maybe it would know. Where is the energy where is the energy for the photolysis reaction coming from? So after it loses the electron, the protein, chlorophyll, becomes extremely oxidizing, one of the strongest biological oxidants known. Okay, so it becomes extremely oxidizing, and so the thing that actually splits the water, the OEC it's called, uses the oxidizing power to pull the electrons from the water. So that's how essentially the water turns into 4H plus ions and 4 electrons, and that electron goes into your boy. This is called an endergonic or endergonic. Let's see, hold on. Endergonic. So this endergonic process under normal conditions, but it's under quote unquote normal conditions, but that strong oxidization that makes that reaction spontaneous, and it's like an abnormally strong oxidization power. Okay, and the redox potential of P70, which is PS2, is not as high as P680, because again, they have different wavelengths of light. And so those electrons that get moved from PS1 have already less energy, so there's less energy actually pushing. So since the electrons are removed with less energy, the oxidation power of that hole that's left, or of the hole of the P70, is less, which isn't enough, I suppose, for the H2O to remove its electron and give it to P70. Okay, 0. We're not going to read that because what's the point? Light independent reactions. Okay, for light independent reactions, I think I'm going to use the book because I actually really prefer how they write it. So light independent reactions uses. First of all, is my pen okay? Okay, this is fine, I suppose. Let's use blue instead. That's going to be thicker. Yeah, that's going to be different. Okay, that's fine. Let's zoom in twice. The plot point of oh my god, the plot point, the plot point of Fire Punch is incest. Oh hell no. Can you burn a Luigi board? Can you burn a Luigi board? A Luigi? The buggy part. Luigi part. How is pregnant form? How can you get pregnant? Pregnant. 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 Uh, uh. Alright, moving on. Sorry about that. Not even sure how I got there. Anyways. Okay. So you use your reduced NADP and ATP produced by the light independent reactions to make carbohydrates. This stage, known as Calvin cycle. So you have a series of reactions known as Calvin cycle, and it occurs in the stroma because you don't need. There's no chlorophyll in the stroma. a series of small steps in the reduction of carbon dioxide from the air leading to the synthesis of carbon dioxide. It is a series of small steps resulting in the reduction of carbon dioxide. So this is the main thing. You're reducing carbon dioxide from the air, leading to the synthesis of carbohydrates. Okay? So you're using the CO. So before this, we haven't even talked about the CO2, by the way. Now we're actually bringing in the CO2 into the equation, and that's obviously coming from the air, as well as the H2O that's coming from the roots, right? So finally we've brought in the H2O, we've done photolysis to it, and now we've turned it into NADPH, and now finally we're bringing the CO2 from the air, and that's going to be both being used to make your GALP, which is going to be used to make your glucose, right? Which is going to be used to make your energy. Oh dear. Okay, so first step is carbon dioxide, immediately carbon dioxide from air, carbon dioxide from air combines with 5-carbon compound enzyme, by the way, ribulose biphosphate, RUBP. Okay, so remember, this is not Rubisco. This is not Rubisco yet. Rubisco is okay. Sorry. It needs Rubisco. Sorry, so sorry. That's Rubisco. That's gonna be your enzyme. This is not an enzyme. This is the okay. So this is the enzyme which allows it to occur. The CO2 binds with this and goes through this to form your this. Okay. So you have a 5-carbon compound, and it needs to be fixed into there via carbon fixation. However, it needs your Rubisco in order to be fixed. Okay, so Rubisco is what allows this to happen, and so the result between the RUBP and carbon is a 6-carbon compound, and this exists, but it's highly unstable and has never been isolated. It immediately separates into, so it's almost like an intermediate. Okay, so you have the CO2 plus RUBP, forming that intermediate, which is 6-carbon, via Rubisco. Okay, and it immediately breaks down into your GP, which is your 3GP, which is going to be a 3-carbon. So you're going to have 2 times 3GP, so 2 molecules of GP. And then immediately GP is then reduced. Okay, not immediately, but the GP is then reduced using your hydrogen that's added from, so reduced, which means hydrogen added, hydrogen coming from your NADPH. That hydrogen is going to be what's added to form your GALP, a 3-carbon sugar. What was it before? A 3-carbon compound. So now remember, this is the main significance between these two. This is a compound. This is a sugar. Okay? And what do you know sugars do? They give energy. That's the main thing. 3GP is never in the state where you can break it down and get energy. However, this GALP sugar can. Okay? However, GALP is not the perfect form for it. It can go into a different form, and that form can be even better and more efficient for being broken down into energy. As I said, it comes from NADP, and the energy required for this conversion comes from ATP, and both of these are going to be produced in your LIR stage, LDR stage. Most of the 3-carbon GALP has three. Okay, so it has 3 uses. One use is to replace the RUBP. Second use is to synthesize the glucose, and third use is to be directly moved into the glycolysis pathway to form your other molecules needed for the plant. These reactions occur in both light and dark, and these reactions only stop when no product of light reaction remains, as to say NAD or ATP is not available anymore. And so these reactions of Calvin's cycle are summarized in figure E. Now remember, NAD, NAD plus come from your LIR stage when it's done. Now you can ask, where do these where does this electron carrier come from in the first place? I actually have no idea. But yeah. So there you have it. Pretty easy. It's not that hard. Simplified version. Simplified indeed. Okay, so here they were talking a bit about, man, how am I still not done with this, man, dude? Okay. We're like literally just 2 pages done. And we're not going to obviously talk about limiting factors from the textbook. We're just going to be, actually, honestly, we're done. The only thing we're going to look at is this, what is this called? They talk about one thing, right? Gluconeogenesis. So GALP used in cellular respiration because it can form so many different things, right? However, it can go back to the 4-carbon. Oh, wow. What? So GALP can be turned back into RUBP. GALP can turn to a 4-carbon rather than a 5-carbon. Wait, what is GALP? It's a 3-carbon sugar, so it can turn to 4-carbon, but where does that carbon come from? Okay, it's just some carbon mixing, right? So it turns into 4-carbon, used into nucleotides via the phosphates from the soil to form nucleic acids. Remember the most important ones. You have your carbohydrates, you have your nucleic acids, you have your proteins, I believe, and you have your lipids. So you can make lipids, you can make proteins, you can make nucleic acids, and you can make carbohydrates. They don't mention that though for some reason. They do mention sucrose, which is fine, I guess. Sucrose is just I don't quite remember. Is sucrose the is that the dimer? The sucrose dimer or whatever. What's it? What do you call polysaccharides? Is dimer is sucrose a disaccharide? Yes, it is a disaccharide composed of glucose and fructose subunits, and fructose is probably a monosaccharide. Fructose monosaccharide or disaccharide? It is a monosaccharide, a free fructose, or a